is the presenter for today, and he is a co-founder and CSO of OspreyBio founder and CSO of Interxon, now Presentine. So please enjoy, and let's learn today about what he has to show us.
Thank you, Horacio.
Thank you for coming.
Good afternoon. As Horacio shared, my name is Tom Reed, and I want to...
Sorry. He is there. He's still my current collaborator, so I forgot about that.
Yeah.
Welcome.
Thank you, Horacio.
Wait. Social media or like you're doing a documentary?
I could do both.
All of the above. Okay. No. So to that end, I was right about to introduce Zev. Zev is here, and he's a part of our consulting team to assist with getting our social media message out. I want to make you aware that we're not allowed to record any of your questions and answers during the talk. So I can only record my answer to you. But after today's discussion, if you're open to having some little one-on-one question and answers, we have talent release forms here. So if you want to be on social media, we're ready to have some little Q&As, and we'll try and get those little snippets in. But I just wanted to let you know that we're operating according to all proper legal activities. And with that, Tom Reed, I am the founder and CEO of a company called BioSolution Designs, which is a parent company that has several subsidiaries under it. I will be introducing you to those briefly, but I believe it's always important to share with team members the actual agenda for our talk. There's a lot of material here. There are certain things I'm going to slow down and certain things I'm going to go, and this, and if you want more details, we can share the presentation with you. So today's topics, we're going to have an introduction to myself and my team, and we're going to talk through the drug discovery and development process, because today we're looking at the application of multigenic therapies in the next generation of advanced therapies. And it's important to understand where we all fit within the end-to-end drug discovery and development process. Then I'm going to try and make a case for multigenic therapies, because I've been shocked. We launched OspreyBio officially at the ASCB meeting and EMBO meeting in December of last year. And when I say we have all these tools for multigenic therapies, people are like, what's a multigene? And it's just blowing my mind, because we know that there are complex diseases, and now I'm discovering I have to talk with people about the need for multigenes, and I hope I can do that, and then build the case that the bird of prey platform is the multigenic standard. And then I'm going to quickly go through how you can use the bird of prey either through OspreyBio or potentially pursuing a collaborative endeavor, a collaborative drug development endeavor with our gene and cell therapy subsidiary Multigenics. And then we'll have the Q&A session. So now for the introduction. So you'll know that we're going from step to step as I go from the highlighted. So again, Tom Reed, and I started my entrepreneurial journey as a graduate student at the University of Cincinnati, where in 1998, as a graduate student, I started a company called Genomatics USA. And the focus of that company was to build complex DNA molecules for the genome engineering of transgenic animals. So if you know about knockout animals, knock-in animals, making reporter systems, making overexpression of mutations to see how they operate, that was my specialty. And so I was all about building the hardest of the hardest of the hardest DNA. And then along the path, I started discovering that there was a need for a new set of research tools that will empower researchers to start understanding cell signaling at great detail. And it was at that point when I moved my company from Cincinnati, Ohio to Blacksburg, Virginia, in exchange for a $1 million economic development investment. And then as a result of that, in 2006, I was discovered by a serial biotech entrepreneur who started investing a lot of money into my company, and that company was renamed at that period Intrexon. And that company went from being a transgene services and tools company to where I was forced to shut down all of this and become an all gene therapy, all oncology company. And then 3 years later, the CEO of that company, RJ Kirk, discovered the magnitude of the underlying technologies that we had, and that's when he started pouring on the gasoline. And we expanded all of our health programs and then started entering into food, energy, environment, and consumer. And we had the largest biotech IPO in history in August of 2013. And then we discovered about how the real world works, and we went from a $7 billion company down to a $200 million company over a certain period of time. And the company had to rebuild itself. And in 2020, it became Precigen, and it focused solely on immuno-oncology initiatives in the gene and cell therapy space. And RJ Kirk, through his private equity group, acquired all of the assets in food, energy, environment, and consumer. So really cool stuff is going on. That's another discussion. But today, the goal is to make you aware of what you can do with multigenic systems in therapeutic development. And so this is just a brief overview of, in the height of Intrexon, we were operating in numerous expression hosts, from bacteria to finfish to mosquitoes to fruiting trees. So we were the leading genetic modification company in the world. And a lot of those products are still progressing, but we brought in a series of technology platforms that we had to integrate together so that we could apply them to develop complex products. And in the human therapeutic space, my team and I contributed to all of these different therapeutic candidates, of which one we expect to be approved this year it's a cancer vaccine for the treatment of cancers starting from HPV. But the important thing is, everything that I learned from 1998 until 2020 helped shape my mind about the types of platforms that are needed for the future of gene and cell therapy. And today I hope to convince you that BioSolution Designs and its subsidiaries are on that path. So I retired from Intrexon in February of 2020 and started BioSolution Designs in April of 2020. And it is an invention studio where we focus on the creation of platforms for the next generation of therapies. We also have OspreyBio, which is our tools and services subsidiary that empowers the global research community to use tools to create multigenic systems. And then we have another subsidiary called Multigenics, and this is a subsidiary that has received an exclusive license to all BSD technologies for human gene and cell therapy. And so we currently have programs running in cardiovascular, dermatological, neurodegeneration, and musculoskeletal. And we're always looking for partners to work on accelerating our own programs and or looking at how can we help accelerate your programs with our technologies. So now I'm going to just give a brief, my take on the drug discovery and development process. Everybody has their own view. But I want to start out and remind you that we're all, for the most part, funded by NIH. I know that during my graduate career and all the way through working on development and working with universities, if it weren't for the NIH budget, we just wouldn't be able to do the cool things that we're doing. But I want to remind you what the NIH mission is, and that is to seek fundamental knowledge about the nature and behavior of living systems and the application of that knowledge to enhance health, lengthen life, and reduce illness and disability, okay? So I would argue that that mission because we're getting money from the US taxpayers to do good things for the US and the globe, this is our mission. And another way of defining our mission is to break it into two parts: seek and apply. So when I entered science way back in 1984 as an undergrad at UC Davis, it was all discovery. I mean, we didn't know what biology was doing. The human genome had not yet been sequenced. We were stumbling away trying to find out how one gene at a time worked, okay? But then in 2002, the human genome was sequenced, and then all of these new technology platforms are getting more powerful, and a ton of data is available to us that now we can apply that information. So now we need to be in therapy invention and development. Now, I understand that we always have a target effector discovery action that we have to be doing, but I would challenge you, if right now you're in that basic research activity, be thinking about how you would use that knowledge to invent a therapy. And our goal is to say, the platforms from Osprey will enable you to start designing and building therapies today. So you've all heard of the bench to bedside process. And what I've done is, this is a very busy slide, but basic research falls into defining gene function, understanding the epidemiology, and then developing enabling technologies to further expand your knowledge base and or develop new applications. And with all of this information, you move into target discovery, target validation, effector discovery, and effector validation. And then moving into what I call effector optimization, translational research, preclinical, and then you're moving into clinical. So this is a very long process, but I believe that if you're thinking about the end in the beginning, you can shrink the time to get to a drug. So I'm not going to go through all of these, but again, I'm going to make this presentation available to you. But I am convinced that if you can answer every one of these questions, you will be able to find the optimal drug candidate. Okay? So all of these questions need to have a system that allows you to fit your answer into the actual programming of DNA for the genetic manipulation of cells. So the other thing I'd like to call out is we're leaving the era of single target effector dynamics. Okay, once upon a time, trying to find one target and one effector, a small molecule, trying to find an inhibitor or an activator. And it's because everybody was finding one target at a time and then one effector at a time. We're now in the gene and cell therapy space. Everybody's still doing that one target, one effector in the rare diseases. And the goal is to start getting people to think beyond one target and one effector for complex diseases. So what I like to say is you need to start designing coordinated, multimodal, and multigenic therapies. Okay, and when I talk about multimodal, you want to look at using small molecules, recombinant proteins like antibodies, and gene therapy and genetically modified cell therapy in a coordinated fashion. Now, any of those of you who've ever interfaced with the FDA are gonna, oh, how on earth are you gonna get a coordinated or combination therapy approved? Well, you're not, okay? And so what you need to do is build a map of what a coordinated therapy would look like, and then you work with the agency on getting an approval endpoint for each of the individual elements that then can come together in a coordinated fashion. So this is a very busy slide, but I want to remind you that multimodal therapy examples exist. They're everywhere. And when you look at the small molecule combinations for HIV, today, there are 2 drugs that have a combination of 3 to 5 individual therapeutic effectors in one drug, okay? So the FDA has approved one pill that has multiple effectors in it, but it's taken 40 years for us to get to that level of precision that the FDA will allow us to have this multiple effector therapy in one pill. Okay, so it's multiple different small molecules, but in one pill. And then when you look at the oncology space, we know that there are these checkpoint inhibitors that help improve the effect of immunotherapies to attack cancer. And I'm calling out here just one class of monoclonal antibodies is being used in all of these different types of combination therapies. Okay, so I'm going to ask you to just step back and say, well, when you look at small molecule combination therapies in infectious diseases like HIV, there's a precedent. When you look at oncology, there's a precedent for multimodal, but when you look at other diseases, there's no precedent yet, except one that I established and I'm going to share with you. So what I want to do now is build the case for advanced multigenic therapies. Now, you've probably heard the term advanced therapies, and that's frequently used for gene and cell therapy, but I like to talk about advanced multigenic therapies because I need more than one effector acting on more than one target so that I can have a systemic effect rather than a single local regional effect. And this is a great slide. I wish I could take credit for it, but it's from NIH. And so when you think about monogenic diseases, you have one DNA variant leading on one chromosome from mom and one chromosome from dad, leading to a rare disease. And theoretically, all I have to do is replace that one gene and I can fix it, okay? We all know that there are certain diseases that you have to have correction from inside the cell. And so if I want to add a protein into the bloodstream, that protein's never gonna get inside the cell. So we actually need to have gene therapies that will go into the cell and act from within, okay? So right now, the industry is very excited about rare diseases, and there are 35 approved drugs at this moment in the gene and cell therapy space. This is significant because does anybody here know the year the first FDA-approved gene therapy came online?
I don't remember the year, I know the condition.
Yeah, so it was 2017. So it's not even a decade ago. So this is a new thing. This is an entirely new industry, and I would argue that the industry's reality is defined by the FDA. So people were working on gene and cell therapy since the 80s, but it's just a pipe dream until the FDA approves something. So the industry did not come into existence, in my mind, until 2017. So we're at the beginning. This is an exciting time for you to lead the future, and my challenge is you need to be thinking about complex multigenic diseases. So this is a map of coronary artery disease. So in today's world of using an adeno-associated virus with a payload capacity of 4.2 kb, that'll carry one gene, okay? Do you think one AAV is gonna answer this problem? I would argue no, okay? One AAV may answer a problem like spinal muscular atrophy, which is a terrible disease, and that was approved, but that's just one gene. So when I look at the gene and cell therapy sector, we're predominated by a monogenic trend. And I would argue that we need to be looking at the future because the vast majority of diseases are complex in nature. They're multigenic in nature. Kim.
So wouldn't the monogenic trend be because the FDA process to getting through clinical trials is the inhibiting factor? Because people will just pick one thing?
It's part of the problem. Okay, it's part of the problem.
I know nothing.
No. So your question is outstanding because why do we have this monogenic trend? And I would argue the monogenic trend matches big pharma's understanding of drugs. So big pharma is always, remember their small molecule history. What's my target? What's my effector? So if I've got a target and I got a small molecule, I can explain that target effector biology really well. And I as big pharma can go to the FDA and say, look, I've got all this information about this target. Here's my effector, and here's all my data looking for off-target negative events from my effector. Okay, so it makes sense. There's a paradigm for a single target and a single effector mentality when going in big pharma and going to the FDA. And that's why we're here, okay?
So the corollary to that would be it's cheaper for big pharma to do the monogenic way than it is to do multigenic. Would that?
Absolutely, absolutely, unless you have bird of prey, but we'll talk about how.
It seems like you can't get there from here, but now we're going to be saved by a bird of prey.
Exactly, we're decloaking. But I want to remind you, this is where the biggest needs are, okay? When we think about a healthy life and a great quality of life and a longevity, remember the mission: improving longevity, improving health, and decreasing illness and disability, okay? We need to do something more, okay? And so now, to the impact. So right now, there are over 10,000 identified rare diseases, and those rare diseases have a prevalence of 1 to 5,000 patients per disease. And so all in, there are around 100,000 Americans who die every year from a rare disease. But then if we step over here and we look at the complex diseases, these are the diseases that are killing more than 2 million people a year. And when you look at the prevalence is massive, okay? So there was, one of the things you'll discover about how business works and Wall Street works and venture capitalists work, they'll go wherever they see a precedent established. So there was a company called Avexis that was purchased by a European pharma for $9 billion. That company had one AAV encoding SMN to treat spinal muscular atrophy. $9 billion. Okay, so all of a sudden, all the VCs said, look, we want to make $9 billion too. And they went into every university that had any AAV asset and said, can I license that? I want to start a company. Can I license that? Can I start a company? Because they want to be the next $9 billion sale to big pharma. And the problem is, there was nobody in the space, and then there was a glut. Because we all know how easy it is to make an AAV, right? I mean, it's not easy to manufacture an AAV, but the ability to conceptualize of an AAV therapy and the ability to synthesize that DNA is easy, okay? So what happens is sometimes when there's a new precedent established, people will go after that which is easy, and that leads to a glut, okay? And so I'm gonna give you another example of a glut. So this is my admonition to all of you. Avoid herd mentality, okay? Because what happens is, I was once told that surfers know that in order to catch a wave, you gotta be in the water, okay? And that's why I encourage all of you to be in the water thinking about advanced therapies so that when things start hitting, you're there and you're not sitting on the beach going, gosh, there's a wave. And the waves, you're never gonna get the wave if you're on the beach, okay? And I would argue that what you're seeing a lot of academicians and small biotechs doing is they see other people working on CAR-T, or they see other people working on rare disease, and this becomes the focus. So this is data from the American Society for Gene and Cell Therapy, and they're telling us of all of the gene therapies last year, 55% were oncology and 45% were rare disease. Okay, the one thing I'm not showing you is in the oncology space, 80% of those were all going after CD19-based cancers. Okay, think about that. So everybody on oncology is everybody in gene therapy is in oncology, and of that number, 80% of them are all going after CD19 CAR Ts. So there's no way you're going to differentiate yourself. And the world doesn't need 20 CAR T therapies for CD19. They need 2, 3 that are good. So experientially, I'm a cardiovascular geneticist by training, developmental biology, and I had the honor of being trained for 2 years in a pharmacology and cell biophysics department before moving over to developmental biology. So I was blessed to learn target effector biology, and it's stuck, okay? And so I understand that heart failure is a multi-axial problem. There are multiple axes, and you can't just address one. You have to address multiple axes simultaneously, okay? So in this slide, what I've shown is every gene therapy that has been in the clinic to date for heart failure. None of them have succeeded. Going all the way back to 90s until now, no cardiac gene therapy has succeeded. Okay, I will say, Dr. Hajar and Dr. Cranius have made the most progress. Circuit 2 made it all the way to a phase 3 before it failed, and they're just starting a new one into a phase 2. I'm not sure if it's going to work or not. I don't think it is, but because it's only one thing. But what I want to call out is my company set the precedent for working with the FDA to take a 3-effector therapy into human beings to try and deal with the complexities. And so when we went to the FDA, and this is where I'd like to dispel the notion that the FDA is a non-collaborative entity. It's a very collaborative entity if you work with them versus if you pummel them, okay? So there's a process called a pre-IND, a pre-IND, and then an IND meeting that you can have with the agency where you're making them aware of this clinical program you're going to bring to them. My strong admonition to you is get a pre-IND meeting as soon as you can so you're making them aware of all of the details so you're not freaking them out. Because during my pre-IND meeting, I said, I want to take 5 genes to treat heart failure, and they're like, you're crazy. We're not going to let you take 5 genes into a human. But we worked with them to get it down to, okay, we'll let you take these 3 genes in, and if these 3 genes work, we'll let you put a 4th in. And then it's going to be up to you to decide whether you want to take the 3 gene all the way through the process, or you want to go back and start at 0 and do the 4 gene. But we had the precedent established with them, and I'm pleased to say that we had these 3 genes, VEGF-A165 to deal with revascularization, we had SDF1 to promote stem cell homing to the heart, and we had S100A1 to improve the cellular genetics. We took that into the clinic successfully, made it out of the clinic, and with just 1 dose of naked plasmid DNA given via retrograde coronary sinus infusion, 50% of the patients showed improvement. Again, 1 dose of naked plasmid DNA that's not going to be there very long because it's going to be episomal and it's gone, still had a profound effect. And so I retired from Intrexon right as that data was coming out. And that, of course, is right when COVID hit, and people were less interested in gene therapy when COVID hit. So I hope I've made the case for the need of multigenic therapies and or the potential of the need for multimodal systems. Is there a question? Hit me.
Before you proceed, I want to get this clarification. Can you hear me well?
Yeah.
So I believe that 15, 16 years ago, when they approved multi-kinase inhibitors, they kind of paved the path for multigenic approach.
Right, yes.
Since you mentioned about using small molecules for attacking multiple targets, and later on you are kind of talking more about gene therapy approaches. So which one would be fitting into your description of multigenic approach?
So multigenic is for gene therapy. Multimodal is be thinking about whatever you may need to add to your gene, because I think it's a mistake. And what happens is, I'm in the physiology department, and therefore the only thing that's important is physiology. I'm in the cell biology department, and the only thing that's important is cell biology. I'm in the small molecule synthesis department, and the only thing that matters is small molecule. We know that it's ridiculous. Everybody here has something of value to bring to the table, and diseases are too complex for us not to be working together. So to answer your question more specifically, we're going to need small molecules, we're going to need recombinant proteins, we're going to need RNA therapies, we're going to need genetically modified cell therapies, and we're going to need direct gene therapies. We're going to need all of it, okay? And that's why I'm excited about the academic milieu, because you have the ability to start talking with one another about, hey, here's a complex disease, my specialty is small molecule inhibitors, your specialty is recombinant protein production, your specialty is gene engineering for genetic modification of cells. Let's look at how do we make a multimodal coordinated therapy. Does that kind of answer your question? Yes. Kind of. So from my perspective, you're going to see I keep saying GCT for gene and cell therapy. So the challenges for GCTs is, can you build the DNA? And we would argue that there's no complex therapy building system platform available, so that even if you could conceptualize a complex DNA, could you build it? We believe that there are limited controllers to control the effectors, the therapeutic effectors, where, when, and what level are these effectors are expressed. And there's a dramatic inadequacy in delivery platforms, okay? And so these, from my perspective, are the three major unmet needs. And so there's a need for standardized platforms. And so Bird of Prey is our standardized platform that allows you to design and build complex DNA molecules. And we are building into our libraries of genetic componentry that fit our standard controllers and effectors. And we're both discovering new gene delivery systems, as well as making sure that the underlying DNA architecture will allow to accept those genetic modules. So from our perspective, we have the new standard in complex DNA design and construction. And so we have the ability to have a platform for a mono-effector, or we can build multi-effectors, so you can use irises, internal ribosome entry sites, to have more than one open reading frame per gene unit. And we have the ability to have multigenics, so that we can have more than one gene unit. And I'll go into the description of the elements, but we're also building a proprietary set of therapeutic controllers, so that we can control the where, when, and what level our therapeutic effectors are having action. And we at BSD are investing a lot of time and energy into next generation delivery platforms, both non-viral and viral. So from the non-viral perspective, we're looking at how do I improve getting things into the nucleus of cells. And on the viral level, we're going all in on gutless adenovirus. And I can explain the reason for that. But we have systems now that allow you to plug into the current delivery platforms, which I think are less than stellar. But we're going to help you have better control over the current delivery platforms. So we use the term transcription unit to define a transcription regulatory element, an effector domain, an effector linker, and a transcription processing element. So you can think of, some people will call this a promoter, but we call it a transcription regulatory element because we have DNA-based components and RNA-based components all in the regulatory element. And some people would call this a poly-A. There's a lot more that needs to go on in the 3' end of a chain. So our transcription processing elements have RNA-based motifs and DNA-based motifs. And so we break all of these genetic modules into key components that you can readily swap. And we have the ability to take this one transcription unit that fits here. We have a transcription unit linker, and that unit linker allows us to keep adding new transcription units to it, okay? But the really cool thing is, because this transcription unit linker is maintained every time you have one transcription unit, you're moving another one into the linker. The one you moved into that linker carries a linker. So you're always bringing a linker with you so you can keep adding. And the beautiful thing is you can rapidly go from a monogene to a bigene to a quad gene. And if you have 2 quad genes, you'd have an octogene. And that's one of the beautiful parts of having a standardized system for the design and construction of multi-effector multigenics. Now, we have several backbone options, okay? So we have a lot of work in the transient transfection space. So we have a supercharged CMV promoter with a novel intron in it. So it's if you need a transient transfection, this thing's gonna fly for you. But we also have the iris shuttles that allow us to make polycistronic effectors, so more than one open reading frame. And so with our system, we've proven that we can get efficiently 3 open reading frames per effector domain. And then over here, we have an in vitro transcription backbone, and we have a series of genome delivery backbones. So if you want non-viral transposons or recombinases, we have that, and we have viral construction shuttle vectors as well. So now you heard me talk about our R is not just a promoter and our P is not just a poly A. We have built the bird of prey around natural gene architecture. So if you were to blow up a picture of the double-stranded DNA around a gene, it's full of transcription factor binding sites, it's full of splicing sites, it's full of all of these controllers and effector elements. And so we need to understand, how do we engineer the biology the way we want it versus just taking what nature gives us? Okay, so back in my day when I was making transgenic animals to understand transcription, I had 9 kb promoters that I would put into a transgenic animal to try and replicate native genome function. And I just took what nature gave me. Okay, now we have the ability to start making hybrids of nature plus a little supercharging all the way to totally synthetic. But whenever you move away from nature to fully synthetic, you may have off-target effects. And that's why it's really important to have a standard that allows you to start constructing and testing based upon a standard. So again, the bird of prey platform allows you to have every DNA and RNA encoded element and protein amino acid encoded elements tracked, so you can design and build for all of this complexity. And so we say the REP, the R has DNA elements and RNA elements. The E is where you have irises and ORFs and bioactive RNA, and the P is where you have RNA plus DNA. So in brief, the way we have standardized, we're fully taking advantage of the era of de novo synthesis. And I want to remind you that people will tell you, you'll read in genetic engineering news, that somebody has developed a new de novo synthesis platform that can do 7 kb in a certain period of time. I'm the biggest customer of every de novo synthesis provider for that which is impossible to make. So when you go to Twist and you go to IDT and you want to order something, you'll frequently get a rejection notice. Okay, well, I'm the company that gets the most rejection notices because the things I want to synthesize are complex. I'm interested in the regulatory elements. I'm not interested in the open reading frames that I can just change the code on to get away from some hairpin or something. I need all of those GC-rich regions and those hairpins and all that. I need that, right? Because that's where the function comes. And so I have to go to different vendors to use different de novo synthesis technologies based upon the complexity of the DNA that I'm ordering, okay? But the beautiful thing is, once I have that piece of DNA and it's standardized to the bird of prey, I can build it over and over and over, okay? So whenever you try and do de novo synthesis, you may or may not be able to have it synthesized. But once I have a mod, you'll always be able to use that mod in a construction process. Okay, so what we've done is we've chosen a series of rare restriction enzyme sites. Okay, and so these are restriction enzyme sites that are 6 to 8 base pairs in length and occur very infrequently in the mammalian genome. And the reason why we chose rare sites is whenever we build a new genetic module, we have to remove those sites from within the genetic module so that I can maintain my linking architecture. So our linking architecture is based upon the use of NEB's high fidelity enzymes that all operate in one buffer at 37 degrees. So you don't have to worry, well, I gotta cut this one, clean it, do something else. Everything works in the same tube at the same time. And so the R domain is flanked by SAL and PV1. The E is PV1 and Cas. The P is Cas age. And then this is really cool. The TUL is flanked by ZO and NOT1. So whenever I want to move a transcription unit into a transcription unit linker, this SAL goes to this ZO, and a NOT goes to a NOT, and the SAL and the ZO destroy each other. So now what I've done is I've created a bi gene that I can then pick that bi gene up at SAL and NOT, and I can put it into another transcription unit linker at ZO NOT, and just keep going. And so when we talk about botbizing genetic componentry, we have to take restriction enzymes out so that we have a module that has only our restriction sites at the linking points. And so this is just showing you, I'm not going to go into all the details, but this is just showing you that we can make a multi-effector. So here we have our iris shuttle vector. And so we have RFP and GFP with an iris. And then we can move a second ORF in here at PVU1 goes to PAC1. That's a compatible cohesive end. They destroy each other. And now you can swap in this ORF1 at PVU1 and SCA1. And so SCA and EcoR5 are blunt, and they destroy each other. So now you have a multi-effector where you have two open reading frames with an iris in between. And if you're so interested, you can actually build a third or a fourth or a fifth. But we've found that the maximum for really efficiency is no more than 3 ORFs because the translation efficiency drops across every iris. And this again is just showing you SAL goes to ZO, NOT goes to NOT. SAL and ZO destroy each other, and now I have a bi gene that I can grab with SAL and NOT. And I'm gonna just skip this. I will say, whenever you're bored, we can go over, but what we did was we built a quad gene library where we took a series of effector reporter genes, put them into a library of transcription units, and then built a monogene, built a bi gene, and built a quad gene. But I hope you understand that on day one, you can build a library of transcription units that you're then linking with each other. Now, it's important to note that all of our transcription units have unique sequences in the R and the P. So if you go to a vendor and you want some promoter, that promoter is going to come in a shuttle vector that has the same polyadenylation site. Okay? What happens when you build a multi-gene with the same polyadenylation site? Intravector recombination. It'll destroy itself either in the bacteria or it will destroy itself in your cells. So that's why we build a library of unique transcription regulatory elements, and unique processing elements so that you can build a multi-gene and no sequence greater than 20 base pairs will ever occur in that same vector. And that way you're reducing any intravector recombination. And we've got Horacio and Robin and Renee's baby pictures here. So we have a library of internal ribosome entry sites, and we showed you how the structure operated. And what this is showing is in HEC293 cells, our irises are working, and every one of our irises works, okay? Yay. And we have a library of transcription units. And remember, each of these is a unique sequence, a unique R and a unique P. And you see that the green is showing us that we have different levels of expression in HEC293. And again, Horacio and Renee, thank you for this data. We appreciate it. So now what I want to do is say, hopefully I've convinced you the future is controllable, multimodal, multigenic therapies. And hopefully I've made a case that bird of prey can help you get into that multigenic world. And so if you believe the first and you're interested in the second, this is how you can work with us. And so remember that long list of questions that you need for therapeutic design? The bird of prey platform allows you to start designing therapies to meet all of these questions. And so I tried to break that big line of questions into simple, what are your cells? What are your targets? And what are your candidate effectors? Okay, so when you're mapping out your disease, you got to know what cell are your targets in, because if you have a disease where the target's in every cell, you're going to have to try and get your drug delivered to every cell. If my target is in a cardiomyocyte, then all I got to do is theoretically get it to a cardiomyocyte. Okay, but you need to know what's the target, and what are the cells, and then what are the effectors so I know how to get it there. And so these are all of the design elements, is how do we deal with gene engineering? So gene addition, gene knockdown, deletion, or gene editing. How do I look at the genomic status of my gene therapy? Is it episomal, transient, or stable? Or do I want to integrate it, and what are the routes I want to take to integrate it into the cell? What are my delivery approaches, non-viral or viral? How do I choose where I'm going in the genome? This is just telling me, do I want it in the genome or out of the genome? This is telling me where do I want it in the genome and how do I get there? And then what engineering approaches do I apply to try and make this happen? Okay, so there's a lot of questions, and you need a big toolbox in order to do that, and we're building that toolbox for you. And this is, it's a very simple slide, but I love this slide because when I look at the seek and apply, I believe this slide covers it all, okay? So I need to create a disease model. What are all the ways I can create a disease model? Or do I want to report on the status of a disease? So I make a reporter gene, put it in, and I give a small molecule, and hey, disease is going down, and I've got a reporter telling me that. Or I can create a therapeutic candidate. And this is a slide that I made in 2001 because I was trying to convince people to make transgenic animals where in one piece of DNA they were doing all three of these. So the goal was have an inducible switch that would be able to turn on the therapeutic, an inducible switch that would create the disease, and then have a reporter gene so that you're able to have a report of what's going on in the animal in real time. And so that's where the whole notion of a modular multigenic design and construction system came about. And so how to interface with us, We have OspreyBio. It's our biotools and services company where we encourage you to think multigenic. And we have a series of tools where we have curated controllers and curated therapeutic effectors. Our goal over the next 5 years is to have a catalog of every candidate therapeutic effector, be it a known protein, secreted antibodies, or bioactive RNA-based knockdowns, long non-coding RNA and microRNA. So we are going to keep adding to our therapeutic effector list at the same time that we're adding to our controller list. When we came out for OspreyBio, we thought the world wanted to do it yourself, DIY, and so we were all based upon helping you clone. And this is my 6th lunch and learn since July of 2023. And 70% of the people who I've given this talk to have all said, would you just build it for us? And so I'm pleased to say, you got to understand, I built a factory with 120 people in Blacksburg, Virginia that made 20,000 complex DNA molecules a month. It's a headache. I never wanted to build a factory again. I just wanted to take my cool little platform and license it to the DNA construction companies. There is no DNA construction company other than Vector Builder and GenScript who will even talk to you about complex DNA. I've gone to every provider and nobody wants to work in the complex DNA space. They just want to synthesize elements and pop them in to vectors. So I'm pleased to share, we now have our DNA vector assembly services. We'll have our website up in the next week, and we will be taking orders to build complex DNA that is based upon the bird of prey system. And our goal is, you just tell us what you want. You come in, do you want an RNA in vitro transcription vector? We'll make it for you. Protein production vectors, we'll make it for you. Reporter systems, disease models, and therapy development vectors, we can do it all. Because the bird of prey platform is built around the ability to make any therapeutically or research DNA. We can build anything. And I know it sounds arrogant to say that, but you can build anything with the bird of prey platform. And so where do you start? You can either go to the site, and if you want to do it yourself and you love cloning, then you can start cloning, okay? Or if you want us to build it for you, we'll build it for you. I do want to say the value of the catalog of controllers, every cell is different. And no transcription unit is going to behave the same way in one cell to another cell, and no transcription unit is going to behave the same way episomal, transient, episomal, stable, or integrated. And I want to remind you that where you integrate in the genome matters, because you may end up going to a silent spot, and you may have the best transcription unit around, but if you're moving into a site that gets silenced, that transcription unit won't work. So that's why we think it's very important to maintain a catalog of controllers that you can test in your cell and know what are the controllers that should be employed when building a multi-gene. And that's it. We're at the Q&A. Are there any questions? Yes.
2 questions. Number 1, if we use your bird of prey vectors,
yes,
or curated vectors, what is your intellectual properties to patients?
So our goal is to have a minimal royalty on the front. Like, we know that the heavy lifting is how you use it, but we also know that people can't even begin to contemplate how to design and build a therapy without our system. So we feel that we deserve something, but it's not like this crazy, we want 2% of your final product. That's not how it's gonna roll. And we're now working with people to define, is there some marginal royalty, or is it some kind of fixed fee at different developmental stages? So, but again, we don't want people to be afraid of getting nickeled and dimed with royalties. We want this system getting out there as widely as possible. So we're not looking to make a whole bunch of money off of near-term licenses. We want everybody using the system, and we're more interested in how we partner with people to make the therapies versus I'm going to get some money from the DNA that's enabled by the platform.
Getting that the IP expectations are very reasonable, I would like to ask you the second question. You've talked about partnering with academia. So when you partnered with academia, how does it normally work?
Great question. So sometimes we have ideas of therapies that we want to work on. So we have the DNA design, but we don't have the expertise in the cells in the animals. Okay, so that's an example where we've got IP and we're looking for a partner who will be validating, but also co-inventing on improvements. Okay, so we own the IP, we're coming in, you're helping us, and you're gonna get some piece of the action for helping us rather than, I gotta go start another company and raise 10 million before I can do anything, right? Versus, hey, can we work with you in the university and say, we understand this is our IP, but you guys are great at what you do. Let's do this, and let's really be looking at the next generation of product and the next generation where that mix of who owns the majority changes. The other important thing is because we have the multigenics therapeutic entity, we're in a position to license something from the university and mobilize it. So we become the engine of dealing with the VCs, which you really don't want to do. So did that answer your question?
Yes. And the last question, if you don't mind.
But what if someone else had, their own?
Thank you.
Yeah.
So if you have an idea, but you don't have the DNA, we would develop a collaboration. If you've filed an invention where you've got a provisional patent on file or something, and you want us to help you implement it, then that's a different type of, so there are three types of, we've got IP, we're coming to you to help us validate and optimize. We're doing something together from the beginning, or you've got something and you want us to help you optimize. And the terms for those are different, as you might expect.
Going back to the first model.
Yeah.
If we have a disease and a specific target.
Yes.
And you have the vector, can we start the discussion?
Absolutely. Absolutely. We're excited about that. The other thing that I'm upset with myself for not making the slide, in my former company, Genomatics, I started a thing called the Nucleobank because my old technology was called UltraVector. Bird of prey is logs better than my old technology. But back in the day, I would go to tech transfer offices and say, do your scientists have meaningful controllers and or effectors that you would like me to put into our library, because then we can become the salespeople for your componentry. And so we are revamping that program. And I'm pleased to say West Virginia University and the University of Cincinnati are now bringing in their controller intellectual property so that we can build that into a catalog and make the world, make it available to the world. And again, that's the key of having a standard. So Addgene, amazing not profit, but you have no idea really what you're getting when you go to Addgene. I mean, what level of detail, what's the rigor that went into the material that was transferred to them. With us, all of that is going to be standardized so you know you can use it, right? So if you go to Addgene, am I going to PCR this? Am I going to restriction enzyme that? I don't know. But with us, you know how everything is built. Any other questions? I want to thank you for your time and attention today, and if anybody would like to ask me a one-off question and be filmed for future social media endeavors, please let me know and we can have you sign one of these talent forms. But Robin, Horacio, Renee, thank you very much for creating this forum. Thank you.
Excellent. Thank you so much, Tom. did any of you guys have questions moving forward? And you're like, what was that platform? You know where to find it. You know where my office is. I'll always push your connection. Thanks for attending today, everybody.
Thank you.
Nah.
Let me.
Pick you up.
You know.
Oh, thank you very much.
So we'll keep in touch.
Yeah. I'll look up. I'll send you a note, and you can let me know
Sure. Yeah.
what you're interested in. The other thing I'll do is look at your website, and so I'll get a sense ahead of time. Very good to meet you. Thank you.
Well, I definitely enjoyed it.
Okay, good.
Yes.
Did it make sense?
Yes. Yeah. And it's a little bit different than our typical virtual rares, because those are really dominated by elusive equipment.
Right. Right.
So this was kind of more than it almost a lecture that we missed.
Oh, good.
So I think really, really well done job.
Okay.
Thank you.
Thank you. I appreciate that. Well, this is my sixth one, so
You got some practice on it.
the first one was the worst. It was really bad. So.
That always happens.
Good to see you. How are you doing?
Good. Let's talk.
Yeah.
Really. So it's amazing. I think you can sell the vectors.
We're finally selling.
Oh,
We're finally selling.
good.
Yeah.
So the vector is for promoter. Like for example, you have a GFP. I didn't read it carefully because really fast. So for the GFP and the gene, the dual promoter or share the same promoter.
So every transcription unit has a unique promoter and a unique R and a unique P.
Oh.
Because again, if I build a biogene, I can't have the same sequence occurring more than once, because I'll get intravector recombination. Okay.
Yeah.
So that's why we have these libraries of transcription units, so you can find out which ones work in your cells, and then you know which ones you can link, because you showed that they worked in your cell, and you can link them without injuring each other.
Okay, I see.
Does that make sense?
Yeah, makes sense.
Okay.
I thought you can maybe for GFP and packaging, they can share the same promoter or dual.
Well, again, you can have more than one open reading frame with two irises. So one iris, two ORFs, two irises, three ORFs. So you can share the same promoter for a multi-effector.
Yeah.
But with our system, you can have a multi-effector bi-gene or quad. So again, I can have a transcription unit that has three ORFs, and I can link that to another transcription unit that has three ORFs, as long as the irises are different.
Yeah.
Okay, but that's why I have iris libraries, so that you can find the ones that work well in your cells, and then you can make them so they don't recombine.
Okay, I see.
Okay?
Great
seeing you. Thank you.
Thank you.
Okay.
I should then accept it.
One quick, if anybody has a specific question that you want to ask me and have Zeb film you for posterity, I have these wonderful talent release forms, and I totally get it if you don't want to. Because with that, if nobody has another question, I'm going to turn off the mic and we're good.